Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hello everyone, my name is Bhumika and I'm your host for today's episode. This is the first of a two-part series based on papers published in June in the field of imaging tools for diagnosis. The papers included in this series are mainly reliant on structural changes and connectivity of the brain. They also explore applications of both the EEG and MEG technology. In this episode, we will cover papers pertaining to the prediction, assessment and improvement of imaging methods, and classification categories. I will also introduce a few papers of the detection and diagnosis brain imaging category, which will then be continued in the next episode. As a new addition to the AMiNDR team, this is my first time hosting an episode. I have worked on summarizing these abstracts myself, with guidance from Ellen and Sarah every step of the way. I hope to get to bring a lot more episodes to you in the future. Don't forget that each of our episodes are accompanied by a time-stamped bibliography where you can easily find all the papers we have covered. As our listeners, we welcome any suggestions and feedback you can give us, which we can incorporate to enhance your experience with us in the future. So grab yourself a snack and a drink and let's dive right into it. Bear in mind, I will be referring to Alzheimer's disease as AD a number of times throughout this episode. First, we're going to talk about 5 papers in the prediction category. First up, we have Modifying the Minimum Criteria for Diagnosing Amnestic MCI to Improve Prediction of Brain Atrophy and Progression to Alzheimer's Disease. First author is Vuok Sima, last author is Kremen, and journal is Brain Imaging and Behavior. This study explores the improvement of diagnostic tests for mild cognitive impairment or MCI with the intent to increase the accuracy of the 3-year prediction of brain atrophy and the subsequent onset of Alzheimer's disease. This test was conducted in cognitively normal or CN as well as MCI participants with a minimum requirement of deficits on 2 episodic memory tests. MCI individuals were further classified as having either normal or impaired performance in the Auditory Verbal Learning Test, also known as the AVLT. Baseline brain as well as cerebrospinal fluid biomarkers such as amyloid, tau, and p-tau marked a notable difference between the CN, AVLT negative, and AVLT positive groups. The study found the AVLT positive and CN groups exhibited similar medial temporal trajectories. On the other hand, the AVLT negative group demonstrated substantially greater medial temporal atrophy and higher AD progression rates than the AVLT positive group, a prerequisite of impairment on at least 2 MCI diagnosis tests can enhance prediction of medial temporal atrophy and its conversion to AD. Next up, we have longitudinal survival analysis and 2-group comparison for predicting the progression of mild cognitive impairment to Alzheimer's disease. First author Platero, last author Toba, from the Journal of Neuroscience Methods. The author studied the converter and non-converter MCI subject groups comparison approach and the longitudinal survival analysis approach for the construction of predictive models. The predictive models combined MRI markers with neurophysiological measurements. These markers have been used to follow minute anatomical changes during the evolution of AD. The 2-group comparison approach employed linear discriminant analysis, while the survival analysis approach used a combination of risk ratios and logistic regression models. The proposed approaches were evaluated using Alzheimer's Disease Neuroimaging Initiative with a selected small set of markers. These markers helped create robust and reliable predictive models. The best models made predictions with relatively good accuracy. When considering the patient's longitudinal trajectory, this model showed increased prediction accuracy of conversion to AD compared to other published models. The survival-based model showed better specificity and sensitivity balance than the two-group comparison model. Moving on, this next paper is titled Quantifying Uncertainty in Brain Predicted Age Using Scalar on Image Quantile Regression, looks at methods of brain age prediction. The first author is Palmer and the last author is Nichols from the Neuroimage Journal. Brain age prediction using anatomical MRIs has the potential to indicate brain changes. This is suggestive of various neurodegenerative diseases. This study proposes a penalized functional quantile regression model of brain age of cognitively normal subjects in the Alzheimer's Disease Neuroimaging Initiative. This model overcomes the hurdle of neglected uncertainties in other existing studies using functional data analysis. This model can be used to effectively predict brain age in MCI and AD. As opposed to point predictions of existing machine learning approaches, the proposed model gives a prediction interval for each subject. The next paper is titled Focal White Matter Disruptions Along the Cingulum Tract Explain Cognitive Decline in Amnestic Mild Cognitive Impairment, AMCI. The first author is Gosdas and last author is Hosseini from the Science Reports Journal. Aging and neurodegenerative diseases are often reflected in brain white matter abnormalities. The authors uncover a new method to reliably measure fine-grained changes in white matter properties linked with cognitive and memory decline due to aging and MCI using advanced multi-shell diffusion MRIs. The tract profile changes observed were of parallel nature among amnestic mild cognitive impairment or AMCI and healthy control or HC candidates, with a notable focal shift at particular points along subserving memory tracts in AMCI. AMCI subjects demonstrated remarkable white matter alterations at specific locations in the human brain as compared to HC. Memory and cognitive functioning prediction in AMCI is indicated by observing properties along the cingulum tract. This study concludes that white matter alterations at specific points of the cingulum bundle could be significant in early prediction of AD and cognitive decline in AMCI. Although a little miscategorized, the last paper in this category looks into genes associated with AD. Much like an intruder, this paper managed to make it past us under the disguise of prediction. This paper is called Genome-Wide Association Study of the Rate of Cognitive Decline in Alzheimer's Disease Patients Identifies Novel Genes and Pathways. Let's see what first author Sherva and last author Green of the Alzheimer's and Dementia Journal have to say. This study showcases a genome-wide study to examine the rate of cognitive decline, or ROD, in AD patients. Genetic variants and their interaction with time since diagnosis were tested to predict ROD of a composite cognitive score along with a pathway analysis on the top genes. A set of 4 chromosomes, the intergenic region between chromosomes 4 and 16, as well as contactin-associated protein displayed suggestive associations. Neuronal development and function, apoptosis, memory, and inflammation-associated pathways were recognized. While AD risk variants do not have a significant impact, AD progression was determined by AD pathways, intelligence, and neurological functions. This paper is more pertinent to the Genetic Landscape episode hosted by Marsha. Be sure to check that episode out for more papers like this one. The Assessment or Improvement of Imaging Methods category has just one paper with rather technical forward language, called Dose Reduction in Brain 18F FDG PET MRI. Give it half a chance. First author Soret, last author Cass from the Molecular Imaging and Biology Journal. Evaluation of brain disorders and a reduction of ionizing radiation exposure is achieved using high-sensitivity integrated positron emission tomography, or PET, and magnetic resonance imaging, or MRI systems. This study investigated the potential reduction of activity time by half without degrading the 18F FDG PET exam image. A retrospective simulation of reduced injected activity was conducted in patients previously assessed for cognitive impairment with PET MRI scans. A reference set of the 20-minute images, PET STD, were generated along with simulated low-dose injection 10-minute images, PET-LD, using list mode data acquisition. A blinded visual interpretation was carried out using reproducibility between PET-LD and PET-STD, along with comparisons between suggestive Alzheimer's disease and frontotemporal dementia, or FTD cases. A high intra-operator agreement between the two visual assessments was observed, along with high reproducibility of SUV ratios. Comparisons between AD and FTD metabolic profiles generated similar outcomes with PET-STD and PET-LD. It was concluded that a significant reduction in 18F-FTG dose doesn't adversely impact diagnostic capabilities in a 20-minute PET or MRI scan. Before we dive right into the classification category, this might be a good time to take a few seconds to stretch a little and take a couple of deep, relaxing breaths. Here, we will go over 6 papers, each describing methods to classify subjects as being cognitively normal, AD patients, or having other neurodegenerative diseases. The first 2 studies involve the use of MRI technology. The first paper is titled Reproducible Evaluation of Diffusion MRI Features for Automatic Classification of Patients with Alzheimer's Disease. First author, Wen. Last author, Colliot, of the Neuroinformatics Journal. Diffusion MRI has been used for the automatic classification of Alzheimer's disease, but classification performance of different approaches are challenging to contrast due to variability of components like feature rescaling, or FR, and feature scaling, or FS, among others. The scarce availability of these components makes reproducibility of these studies difficult. This paper is an extension of a previous study by Sampa Gonzalez et al. 2018, which begins by extending previously established framework to diffusion MRI. This involves transformation of diffusion MRI AD-NI data into the BIDS standard and pipelines and applying the framework to compare different components. It was found that FS improves classification results with higher balance accuracy, or BA. Voxel-wise features performed better than regional features. The small data samples could have resulted in poor performance seen in MCI tasks. There were no significant classification differences for varying degrees of smoothing and registration methods. Over-optimistic and unreliable results were obtained when using the non-tested validation of FS. The authors concluded that a proper use of FR and FS lead to diffusion MRI and T1W MRI having comparable performances. Next up, we have development and validation of an interpretable deep learning framework for Alzheimer's disease classification. First author, Q. Last author, Kolachalama, from the journal Brain. This study sheds light upon an interpretable deep learning strategy describing distinctive Alzheimer's disease signatures from multimodal inputs, MRI, age, gender, and mini-mental examination scores. The proposed framework links a convolutional network that generates high-resolution maps of disease probability and visualizations of individual AD risks to accurate diagnosis. The model was developed with cognitively normal and clinically diagnosed AD subjects. It was found that the multimodal inputs-based model performed consistently across all datasets. The approach proposed in this study surpassed the diagnostic performance of a multi-institutionalized team of neurologists, and it predicts high-risk cerebral regions, which tracks post-mortem histopathological findings. This framework yields a strategy for using routinely available imaging techniques like MRI to generate subtle neuroimaging signatures for Alzheimer's disease diagnosis. Machine learning and bioinformatics techniques are key players of the imaging game. The next few papers report the use of these methods. Let's talk about the paper, Stratifying Patients Using Fast Multiple Kernel Learning Framework, Case Studies of Alzheimer's Disease and Cancers. First author, Jiang. Last author, Tran. Journal, BMC Medical Informatics and Decision Making. Predictive patient stratification is growing in significance in order to determine the most suitable intervention with a patient. The diverse and complex nature of medical data has made it challenging to integrate past stratification models and apply them to practical use. Data integration is made challenging because of the complex and lengthy computation due to multiple dimensions of biomedical data as well as the disparity between data types. The authors of this paper initiate a fast multiple kernel learning framework, or FMKLDR. It optimizes equations to calculate matrix chain multiplication and reduce data space dimensions. Comparative evaluations were conducted between the use of the framework in stratification in Alzheimer's disease and cancer patients. It was found that this method successfully classified between AD and non-AD patients as well as different phases of AD patients. When applied to 6 different types of cancers, the framework showed a greater accuracy in integrated dataset-based classification models compared to single data type models. The FMLKDR improves computational costs and accuracy in both AD and cancer patient stratification and increases efficiency of data integration, kernel fusion, and dimension reduction. This next abstract was short and sweet but packed with information. Let's talk about multi-level diffusion tensor imaging classification technique characterizing neurobehavioral disorders. First author, Dalboni Darocha, J.L., last author, Sitaram, from the Brain Imaging and Behavior Journal. This study explores a new method with 3 levels of analysis of diffusion tensor imaging data. The first being a voxel-level analysis of fractional anisotropy in white matter tracks. This was followed by a connection-level analysis based on fiber tracks between particular brain regions. Lastly, a network-level analysis based on connections among brain regions. To identify neural connectivity patterns for diagnostics, machine learning techniques of feature selection, pattern classification, and cross-validation were conducted. The proposed multi-level approach displayed a high average classification accuracy across the board of patients and controls. This multi-level diffusion tensor imaging approach is potentially useful as a clinical diagnostic tool for brain disorders. Next up, we have regularized bagged canonical component analysis for multi-class learning in brain imaging. First author is Sevilla Salcedo. Last author is Stoker from the Bioinformatics Journal. This study suggests an integrated feature selection and extraction approach for multi-class problems to overcome the existing problems of supervised learning algorithms. This method begins with a calculation of the sign consistency of multivariate analysis or MVA projection matrix feature-wise to establish feature relevance. This then provides a matrix which was combined with a hypothesis test to automatically determine the number of selected features. A reduced set of summary components is then generated, which provides a compact data description. The proposed method was evaluated in 2 multi-class brain imaging challenges via the classification of elderly subjects as well as children in multiple classes. Resulting brain images were represented with 30 to 45% of the original features. This information could be further redefined into summary components, providing enhanced interpretation and classification rate improvements. The last paper in this category has a rather wordy title: Evaluation and Prediction of Early Alzheimer's Disease Using a Machine Learning-Based Optimized Combination Feature Set on Gray Matter Volume and Quantitative Susceptibility Mapping. First author is Kim, last author is Zhang, and journal is Current Alzheimer's Research. This study tested the ability of a machine learning model to classify and predict AD based on gray matter volume and quantitative susceptibility mapping, or QSM. If you're not familiar with QSM, much like myself, let's actually find out what it is together. As I'm seeing right now, the NCBI website tells me that it is a recently developed MRI technique for quantifying the spatial distribution of magnetic susceptibility within biological tissues. That's interesting. Moving right along, they tested their machine learning-based optimized combination feature set on a group of cognitively normal, amnestic MCI, and AD patients. They looked at areas of the brain known to develop high iron and amyloid accumulation in AD. They found that looking at gray matter volume of the hippocampus and entorhinal cortex, along with the QSM for hippocampus and pulvinar brain regions, was the most accurate way for the model to distinguish between cognitively normal individuals and AMCI. What about distinguishing between AMCI and AD? gray matter volume of the amygdala, entorhinal cortex, and posterior cingulate cortex, along with QSMs for the hippocampus and pulvinar region, was the best way to distinguish these with the model. The same features were the most accurate in distinguishing between AD and cognitively normal patients too. Check out this paper for more useful information about machine learning methods. It seems that this approach could be a useful new diagnostic tool for AD. The largest category in the imaging theme is, unsurprisingly, detection and diagnosis. In this episode, we will be covering 7 of the 25 abstracts in this category. Without further ado, let's jump right in. This first paper is titled Automated MRI-Based Deep Learning Model for Detection of Alzheimer's Disease Process. First author is Feng, last author is Guo from the International Journal of Neural Systems. The authors of this study aim to evaluate the positive impact of advanced deep learning methods, a novel imaging technique, on the diagnostic precision of AD. Binary and ternary disease classification models were accomplished by applying three-dimensional convolutional neural networks, or 3D-CNN, to magnetic resonance imaging. Deep learning performance was compared across 3D-CNN, 3D-CNN support vector machine, also known as 3D-CNN SVM, and two-dimensional or 2D-CNN models. The resulting data of the study in terms of classification accuracy, sensitivity, and specificity in NC, AD, and MCI subjects indicates an enhanced performance of the 3D-CNN SVM with MRI. Without any prior feature extraction, 3D-CNN SVM shows efficiency and independence from the variability of imaging protocols and scanners. Due to the safety characteristics of 3D-CNN SVM, it can be useful to screen for AD in the general population. This study also holds value in differentiating AD and MCI individuals from normal controls and improves patient care. You will notice that MRI, PET, EEG, and MEG technology make a significant number of appearances in this category. The next paper is The Impact of Atlas-Based MR Attenuation Correction on the Diagnosis of FTG, PET, or MR for Alzheimer's Disease, a simulation study combining multi-center data and ADNI data. First author is Seekine, last author is Warnock from the PLOS-1 journal. This study examined the use of vendor-provided Atlas-based MRAC on FTG, PET, MR in the assessment of Alzheimer's disease via simulated images. The subjects had undergone PET-CT, PET-MR examinations for oncological staging. FTG-PET were extracted from the PET raw data acquired on PET-MR using vendor-provided MRAC and CTAT. A series of simulation steps were then performed. An error map was calculated for each subject, and the error maps were then multiplied with each of the ADNI cases to result in a database. A cumulative t-value was calculated to determine the probability of AD in each image. The diagnostic accuracy for AD and predicting progression from MCI to AD were evaluated and then compared to original ADNI images. The results showed that the accuracy and specificity for discrimination of AD patients from normal controls remained majorly unchanged, barring a lowering in specificity in a few cases. Additionally, there were no changes in sensitivity, specificity, and prediction of progression of MCI to AD. Aside from a minute reduction of sensitivity, FDG-PET-MR based on Atlas-based MRAC shows similar diagnostic accuracy as CT-based methods. It also showed predictive ability as a commercially usable software. This next paper talks about electroencephalography, or EEG, a common neurophysiological biomarker of AD. It's called Quantile Graphs for EEG-based Diagnosis of Alzheimer's Disease. First author, Pineda, last author, Campanharo, from the PLOS-1 journal. EEG is non-invasive, cost-effective, exhibits high temporal resolution, and gives key information about brain dynamics in AD. This study introduces an approach based on using quantile graphs, or QGs, to classify EEG data. QGs have been used to map frequency, amplitude, and correlation of characteristics of a time series into the topological features of a network. The 5 topological network metrics used in this study indicated that the QG model can discern between AD patients and healthy individuals and indicates the channels that have the best discriminating power. Further wave analysis points out that the AD patients of the study show clear symptoms of the disease likely in its stage. The results obtained serve as clear evidence of the efficiency of the QG method of analyzing complex nonlinear signals generated from AD patients by EEGs. The upcoming paper is titled Transcranial Sonography Findings in Alzheimer's Disease, a New Imaging Biomarker. First author, Yilmaz, last author, Berg, of the European Journal of Ultrasound. This paper aimed to study the use of transcranial sonography, or TCS, as an imaging tool for the assessment of medial temporal lobe, or MTL, atrophy, known as MTA. This was done by performing TCS and MRI on Alzheimer's disease patients. MTL structures were measured on the TCS to generate a MTA score in sonography, called the MTAS. MTL was also evaluated on MRI. The ventricle enlargement score in sonography, or VESS, was obtained by combining the temporal horn and the third ventricle. Imaging methods and patient groups were then compared to validate the use of TCS. The results showed a moderate to strong correlation between MRI and TCS regarding specific MTL structures. MTAS and VESS showed significant correlation with hippocampal volume and successfully distinguished AD patients from controls. MTAS additionally predicted group membership in logistic regression. The results encouraged the use of patient-friendly and cost-efficient TCS when MRI cannot be used to assess for MTL atrophy and ventricular enlargement. This next paper, titled Evoked and Induced EEG Oscillations to Visual Targets Reveal a Differential Pattern of Change Along the Spectrum of Cognitive Decline in Alzheimer's Disease, talks about the applications of event-related oscillations, or ERO, as oscillatory responses in the EEG to particular events to potentially track cognitive decline. Let's hear what first author Chule and last author Demirolp of the International Journal of Psychophysiology have to say. EEG is popularly used for the evaluation of neurodegenerative diseases. The authors of this study reanalyzed data from previous studies to look into potential ERO biomarkers to distinguish different stages of cognitive decline along Alzheimer's disease trajectories. Target responses were examined for evoked and total ERO powers in multiple frequency bands using continuous wavelet transformation, or WT, on averaged and single trial data. A permutation test was executed showing significant variance among the test groups of the study. A decline in total delta and theta responses was observed in the MCI stage with further spatial expansion in the AD stage. A significant reduction level of the evoked delta response in the AD stage was observed. Total delta and theta EROs can be efficient for early detection of AD in the MCI stage, with evoked delta responses helping to detect conversion to AD. The next paper in this category is titled Medial Temporal Atrophy Alone is Insufficient to Predict Underlying Alzheimer's Disease Pathology. First author Jeong, last author Lee. Journal, Korean Journal of Family Medicine. The authors of this study examined the values of medial temporal atrophy, or MTA, in order to identify 18F fluorobeta-band PET-proven AD pathology. MRI, 18F fluorodeoxyglucose PET, and 18F fluorobeta-band PET were performed on subjects experiencing cognitive decline at baseline. A multivariable logistic regression analysis was conducted on variables such as age, sex, APOE genotype, and memory composite scores to explore if MTA is indicative of underlying AD pathology. The multivariate logistic regression analysis showed that MTA was not an independent indicator of underlying AD pathology. A significant increase in predictive power of AD-associated cognitive impairment was seen when variables like the APOE genotype and memory composite scores were considered simultaneously. The study concluded that while MTA alone wasn't enough to predict the presence of AD pathology, considering other factors is crucial. The last paper in this category we will be discussing today is spontaneous MEG activity of the cerebral cortex during eyes closed and open discriminates Alzheimer's disease from cognitively normal older adults. First author is Ikeda, last author is Yamada from the Scientific Reports Journal. This study sheds light on the use of magnetoencephalography or MEG for the detection of early-stage Alzheimer's disease. MEG data was analyzed from the early-stage AD group and the cognitively normal group. MEG was performed in both eyes closed or EC and eyes open or EO phases. Specific frequency values in the cerebral 68 region were compared between absolute power between EC and EO, whole cerebral normalization or WCN power between EC and EO, difference of the absolute powers between EC and EO, and WCN value of the EC-EO difference. The results demonstrated significant differences between the groups in the WCN powers during the EO condition and the EC-EO differences, hence making MEG during resting EC and EO an effective tool to distinguish between early-stage AD and NC patients. And that brings us to the end, folks. Thank you so much for tuning in. We hope that this information was useful to you and any research you're working on. Keep a watch out for the second part of this episode, where we continue on with the imaging tools for diagnosis theme. We look forward to having you back.
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and a member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.